Welcome back to the week 2 lecture. This is the second lecture in the week 2 where we will be discussing about the pedigree. We already have had an introduction to pedigree as to how we would construct a pedigree by talking to people, understood all the symbols that we use. We also understood the power of pedigree analysis as to whether it would tell you whether it is a dominant phenotype, recessive phenotype, resulting from a gene that is located on the autosomes, X chromosomes, Y chromosomes and so on. So, now we are going to look at some problems. Let us test your understanding as to how good you are in understanding the pedigree and whether you will be able to talk about some prediction with regard to the genetics, right. And then we will move into what is called as exceptions. It is not all phenotype that you look at are following into one of the 4 or 5 rules, that is autosomal, dominant, recessive and so on. There could be some exceptions and how that might contribute to the complexity in the pedigree. So, first let us look into some examples of the pedigree. This is one example. So, the pedigree given here traces the inheritance of a very rare biochemical disorder in humans. Affected individuals are indicated by the circle what you have seen. So, every individual that shown here filled one are having a particular a disorder. So, the question is the allele for this disorder dominant or recessive, the phenotype resulting from a dominant condition or is a recessive condition. That is your the question that we ask, and it is very easy because we have discussed already that if a phenotype affect both male and female and transmitted by both male and female, and if it is the parents, one of the parents have the affected is also affected, it is likely that it is a autosomal dominant disorder. So, the allele is most likely dominant because if you look into, for example, here then you would find one of the parents affected and likewise anywhere that you go on and so on. So, this is obviously a autosomal dominant disorder. Now is the question. The question here is what is the genotype for the 3 individuals, individual 1, 2 and 3. Can you use this pedigree and in the phenotype that is denoted to arrive at what would be the genotype of the individuals? The answer is yes, we can. So, how do you do this? You pretty much plot the genotype. So, let us start from number 1. This individual must be having the defective allele resulting in the phenotype. Now, for her to show the phenotype, either even one copy is good enough, one copy of the bad allele is good enough, but she also would show the same symptoms if she is homozygous for the dominant allele. So, how do we know whether she is homozygous for the dominant allele or heterozygous for the dominant allele? The answer is you can look into the next generation. The next generation of the 4 children, one of them is normal, and this individual he could be normal only if he had received a normal allele from her mother, because father would contribute one allele, mother has to contribute the other allele because it is autosomal, and obviously this allele should have come from mother, therefore she should be heterozygous for that dominant allele. That helps us to arrive at this individual also must be heterozygous for the dominant allele. Let us look into the next generation. Here this individual is showing the disorder, but what we do not know is genotype. He could be homozygous for the dominant allele or heterozygous for the dominant allele. Now, how do you know whether he is homozygous or heterozygous for the dominant allele? Just the way we are done here, you can look into this generation and you find 2 individuals are asymptomatic, not showing the disorder. That means they should have inherited one normal allele from each of the parent. She obviously contributed one allele and you must have contributed the other allele, therefore, he also should be heterozygous. That brings us to these 2 individuals, and these 2 individuals we really will not be able to tell whether they are homozygous for the dominant allele or heterozygous for the dominant allele, because both are possible, because this capital B can come from here or from here, or both can come from both parents. So, therefore, there is a 50% probability for each one of the individual that they are homozygous for the dominant allele or heterozygous for the dominant allele. So, with that we will be able to tell the genotype of number 1, number 2 and number 3 without any confusion as to what would be the genotype, like shown here. So, this is how you will be able to arrive at a conclusion as to what would be the mode of inheritance, whether an individual is likely to be a carrier or homozygous for a mutant allele if the phenotype that you see is a dominant character. Now, let us go and look into another problem. So, this time assume that you finish the course and you become a genetic counselor, and there is a family comes to you for an advice with regard to the risk of their future children having a particular genetic disease. So, the condition is given below, you can read it on the screen, that a couple, husband and wife, planning to start a family came to you for information. This individual Rakesh was married once before, and he and his first wife had a child who has cystic fibrosis. It is a disease that is really severe, and the person don't survive long. The brother of the current wife, his second wife Pooja, her brother had cystic fibrosis and he passed away. And this condition, cystic fibrosis, is a lethal recessive genetic disorder, and of course they can't have children because they don't survive that long. So, as and when you have a homozygous condition for the recessive allele, it does not go to the next generation normally. So, the advice I wanted to ask was, what is the probability that Rakesh and his second wife Pooja will have a baby with cystic fibrosis, because there is a family history, right. So, the only information that you have is that these two, Rakesh and Pooja, they do not have the disease, they are asymptomatic, they could be carriers, but that is what the risk is. If they are carriers, they are likely to have children in the next generation, the children could be affected. So, how do you really use this information, whatever they narrated, to build a pedigree and predict the genotype and predict again whether their children would have cystic fibrosis. Let us do the practice, okay. So, let us draw the pedigree. So, who is this? We are going to talk about the individual Rakesh. So, here is Rakesh, right. So, he is asymptomatic, he is normal, and his first wife, right, she was normal, and their son had cystic fibrosis, right. Now, he is married again, and this is Pooja, right. Now, they are planning for a children, and we do not know whether if eventually they have a child, whether he or she would have cystic fibrosis. So, what are the other information you have? So, what we know is Pooja's brother passed away because of cystic fibrosis, right. So, this is the information you have, but nobody else is affected. Obviously, for her parents that we have you have interviewed, and they are normal, let us assume that way. Now, how would you draw the genotype? Let us look in here. So, here this individual is affected, and let us say cystic fibrosis is resulting from a recessive condition, that is what the information already given. Let us say A is causing cystic fibrosis. If you are autosomal dominant, you will have cystic fibrosis. If it is capital, or you are heterozygous, then you are wild type. Let us have this information. Now, like, can we put the genotype? Yes, we can. And this guy Rakesh is normal, therefore, you would expect him to be heterozygous for the mutant allele, because he is normal, but his son had the disease. So, you must have contributed one of the bad alleles. Likewise, his first wife. Now, what about Pooja, his second wife? So, let us look into that probability that her brother had cystic fibrosis. That means his genotype should be homozygous for the mutant allele, that is small a, small a. If his genotype is this, then her parents who are normal should be heterozygous, otherwise you would not have gotten the disease. The question is, what would be her genotype, right? She is normal, wild type. Therefore, she cannot be a genotype which is homozygous for the mutant allele. She has to be either heterozygous or homozygous for the wild types. So, how do you know which is possible, which one of these two possible? Because now we have to talk about the probability of this particular kid having cystic fibrosis. So, what you normally do is, in genetics you put the Punnett square, because these are the parents. So, you say two types of gametes, and then you have these possibilities. What we are saying is, this is not possible because she is normal, right? So, she is, the probability that she would be heterozygous, she could be one of the three conditions. That is, she could be, in this combination, that means that she is, this is the probability of her having the heterozygous for, a condition which results in the what do you call a cystic fibrosis. If this is the case, and we know the phenotype, that is, we are talking about a condition, and he is certainly he is having the carrier. He is a carrier because his son, the first son, had cystic fibrosis because of the homozygosity, and obviously the probability is, you can easily calculate this way, which is equal to 1/6 is the probability that this particular kid would have or develop cystic fibrosis. This is the kind of calculation you can tell and predict and then say whether the new generation somebody would have the disease or not. So, this is how the pedigree also would help you to arrive at certain conclusions, right. Let us look into the complications. Pedigree analysis is not always simple. There are many conditions that really do not follow the typical Mendelian segregation in terms of phenotype. There could be problems, right. One problem that often we face in case of dominant disorder is penetrance, meaning that you have what is called as incomplete penetrance, like what is shown here. So, you call it as incomplete penetrance or non-penetrance, meaning you may have the genotype, but you may not show the phenotype. At times, you may have the defective allele, but you are not showing the symptoms, you are normal. It happens because of a complex genetic interactions. Let us not get into that. So, let us look into this family. What is this family? In this family, what is that? You see an autosomal trait or autosomal dominant trait or recessive trait. You can easily see that almost every generation you have affected, and the affected is either male or female, transmitted by both male and female. That means it is autosomal dominant. But what is interesting here is that you do have individuals like here who are affected, but you do not see their parents being affected. But here again you have an affected, but this generation you do not see anybody affected. Likewise, you see an affected, but this generation is not affected. You see an affected, this generation is not affected. So, this is what we call as penetrance, right. It is not fully penetrant. Just having a genotype does not ensure that you would have the phenotype. So, that is what called as penetrance. Now, how do you really calculate penetrance? Because it is very important for you to calculate penetrance. What is the probability that if I have the genotype for this particular phenotype, I would develop the phenotype as well. So, this is an important question that you want to tell, right. So, that you can calculate for a particular phenotype. Let us calculate here. How do you calculate penetrance? You can do that. So, assuming it is a dominant disorder and it is autosomal dominant, I would usually say that the capital A causes this disease. Therefore, this individual is this, and here, here, let us see the segregation. They must have received one of the mutant allele from that generation, and this is married in, meaning she is not related to this family, DNA wise, she is married into this individual. So, assuming that she would be all right, you would expect her genotype to be having the normal allele, wild type homozygous. This A should have come from this individual, but is a carrier, right? We can denote like this, and is not asymptomatic. This A had come from here, and therefore she should have been homozygous for the wild type, and he is normal, therefore homozygous for the wild type, and she is having the dominant allele, should have come from one of the parents. He is married in, therefore his genotype could be this, and she could be heterozygous. We do not know, we can't say, it could be penetrance. Likewise here, if you see, this guy should have been heterozygous, assuming married in, she is wild type. Likewise, he is having the phenotype, therefore you assume his father contributed this bad allele, he is heterozygous. This is how you plot it, right. Now, let us look at how many individuals could, the minimum number could have heterozygous for, minimum number of individuals that are heterozygous for the mutant allele. You can calculate that 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Minimum is 10. How many of them have genotype? Sorry, how many of them have show the phenotype? 1, 2, 3, 4, 5, 6. So, the penetrance is 60%. So, this is how you arrive at the calculation. So, if you could get very similar number for the same disease in a number of families, you can with confidence say the penetrance level. So, that also help you to predict kind of what would be the probability a given kid could have the disease and so on. So, this is how we calculate. This is one of the complications in Mendelian. You would find individuals that do not show symptoms, but yet they are having the genotype. The other factor that influence your pedigree analysis is variable expression, meaning you have a disease allele, but whether or not you would express a phenotype depends on various other factors. And here it is not like penetrance. Penetrance is whether it is a black and white, whether you have the disease or we do not have a disease. But often you have a condition which is in between these two, that is you have the disease or the symptoms, but it may not be that severe. It could be milder in some individuals, it could be very severe in some, it could be moderate, and that also can be denoted in the pedigree like what is shown here. For example, you could use different types of filling. For example, this is a severely affected individual, not that severe, somewhat milder, and these are very mild phenotype. So, you can use different filling in the symbols for individual, whether it is circle or square, to denote the spectrum of phenotype or the symptoms or severity. That would also help you to understand how the disease in terms of its severity runs in the family. So, these are another information that can easily be brought in, which could help in understanding even the other factors that contribute to the disease, right. So, this is another variable. You have other conditions. For example, you may have a disease that is resulting from a particular genetic defect, particular genetic mutation in a gene, but all of a sudden that the phenotype may vanish from an individual. He may have the mutation, but may not show the phenotype. This is called as a complementation. More often it is heard in condition wherein you have hearing loss because of a genetic defect. Let us see like here. You are talking about a genetic defect in two different genes, and here in this family, for example, it is resulting from a defect in, for example, this small a, right. And you have a heterozygous parents and you have defect, and you look at this particular individual who is having the defective allele in homozygous conditions, therefore, you are showing the symptoms, and he is married to an individual again having a very similar condition, and but she is having a defect in altogether a different gene, and that condition is also recessive, and therefore you have both copies in homozygous condition, and she is showing the symptoms. It so happened when in marriage happens between two such individuals having very similar condition but resulting from two different genes, the next generation you would find that their children are normal. It happens because one of the gene defect can mask the effect of other gene defect, right. This is called as complementation, meaning whatever defect that results from here, that is rescued by the other gene defect. On their own they result in disorder, but when they come in combination like this, it rescues. These are known in certain unique cases, and this by looking into the pedigree, only looking at the pedigree, you will be able to tell, you would not anticipate that they are having the genotype. You would assume that they are heterozygous, therefore they are not showing, but there are complications. So you should really look at such kind of cases as well. These are exceptions, but not very common, but it is a rare condition, but certainly there are conditions like this. The other complication in Mendelian pedigree pattern is imprinting disorders, what you call, caused by defects in genes that are normally expressed from one of the two alleles that we receive from parents. These are autosomal obviously, therefore, when you look into the gene copies, one of the copy you received from father, the other one has come from mother, and we know for certain genes only the allele that you received from father or mother would express. So, that is something that is shown here. So, as you can see here that you have gene that is present in both the maternal and paternal chromosomes, and what is shown here is that, for the example, if you look into this lollipop, assuming this is a gene, this gene is expressed only when it is located on the paternal paternal copy, that only that particular gene is expressed. The one that is located on the maternal chromosome is silenced. In some other genes it is the reverse, that is the opposite. So, what would happen in this case is that for such gene, for example, a gene that is normally expressed on the paternal allele, if the mutation for the same gene is present on the chromosome which gets silenced for this particular gene, now this, if an individual having this kind of a genotype where the mutation is present on the maternal copy, then he would still be normal, because anyway this copy is supposed to be silenced. On the other hand, if the mutant allele happen to be the paternal allele, now that although the other copy is wild type, this cannot rescue this defect because this gets silenced. Therefore, this individual would have the disease. So, what you see here is a pedigree wherein the colors that the dots either within the circle or the square which represent the carriers. Now, what we are trying to show here is that how the same defective allele when passed through either mother or father results in having a given disorder or not. So, you can look at here, for example, this individual is a carrier and he also shows the phenotype. So, he has a defective gene, but this he inherited from his father, therefore, you show it as a red color like what you can see here. There is a paternal allele is red color, so he received it from father, therefore, it is red, and that should be expressing, since that is having defect, he end up showing the Prader-Willi syndrome. Likewise, his daughter and his son. However, his father is normal though he is carrying the defective allele, because this particular defective allele has come from his mother, therefore, is maternal, and the maternal copy anyway should be silenced. Since he has got father who has donated the normal allele, he is all right. Likewise, you can see here from mother, this is again it is silent, therefore, it is not affecting her daughter, and again she is able to contribute this mutant allele to the next generation, that is to his son. Again the son is not affected because this is maternal allele and it is silenced anyway, but however when it goes to the next generation, you see that it you see the disease coming up, because this is the paternal allele that should be expressed here, because this goes from a male. Likewise, you see that how a defect, depending on whether it is paternal or maternal, in which lineage it is passing through, would end up developing into a disease depending on the parent of origin effect. So, that is an example of imprinting disorder, and such complications really, when you look into the pedigree, it would be very difficult to understand the segregation pattern unless and only until you know that they are representing imprinting disorder. The other example is called as anticipation. So, if you recall, we have discussed one condition called as variable expression, wherein in every generation it can become severe or less severe or moderate or more severe and so on. But there is one condition in which in every successive generation the disease become more severe and severe. Not only that, the age at which you develop the disorder is much, the age at onset it decreases. For example, in the current generation it could be, age at onset is 12. In my previous generation it could be 20, the generation before that it could be 40, and that is what denoted here. If you can see here, now you have condition, 3 generation family, and you can see here this individual carried a defective allele, and it is a dominant disorder, every generation both male and female affected, and you can see that she developed a disorder at 65, in this generation it is around 40, in next generation it has become around 20. So, this is called as anticipation. So, you can expect in next generation the disease to become more severe and earlier onset, and this happens because of one particular group of mutation called dynamic mutation, which we will be describing later, but this is something very, very unique and exception to a typical Mendelian pattern. Now, we are going to look into some issues with regard to how the mutation happened, when does it happen, and where did it happen, right. You may have a disease all of a sudden in a family, need not be inherited. The previous generation may not have had any symptoms, and this happens because of what is called as de novo mutation, meaning something happening for the first time in a family. So, in a germ cell there was a defect, and as a result you have a genetic mutation, and from next generation onwards you see the symptoms, and such kind of mutation can happen anywhere. It is a random event, right. It can happen in the germ cells, or it can happen in non-germ cell, for example somatic tissue. So, depending on where it had happened, it affects whether it will be inherited or non-inherited. So, for example, if you have what is called as a somatic mutation, mutations happening somewhere in your body other than your germ cells, it may result in certain conditions. For example, many tumors or cancers result from somatic mutation, but they are not transmitted. Such kind of defects are not there in the germ cells, it does not go. But if there are defect happens in one of the germ cells of an individual, and if it so happened that that germ cell led to the formation of embryo and children, then you would see that in next generation onwards you have the phenotype. So, these are called as germline mutations that can be transmitted. But it may so happen that the somatic mutation can have what is called as you have a mutation in your other than germ cells, and then you have group of cells in your tissue, some of them having one particular mutation, other one are mild wild type. So, that is called as mosaicism, right. So, that can result in some individuals having patches of different colors of skin, that is one examples of mosaicism. So, how does it happen? So, there are two different possibilities for this kind of complexity. One is, for example, as shown here, you have sperm and egg fertilizing and resulting in a zygote. As the zygote multiply and form an embryo, there could be one particular cell acquires some mutation, and as the cells divide, and all the cells that are derived from this particular lineage would carry the mutation. So, you would have an individual who is having two different genotype. Some group of cells are, for example, heterozygous for a mutation, the other groups are wild type for that. Or there could be conditions wherein during development you have had two independent embryos resulting from the fusion of two independent sets of sperm and ovum, and it may so happen that when they are developing together, they may fuse together to form what is called as a chimera. They have mixture of two different sort of individuals, so to say, but eventually they develop as a single individual. So, when that happens, you could have variation within a body. For example, in the difference in the eye color that you can see. And there are many such examples people have shown, wherein individuals are, for example, or chimera for X46, XX and XY. There are some cells that are XY, some cells that are XX, and so on. There are conditions. Also, there are individuals that are known to have, blood cells for, two different, blood groups, and so on. And there are many such conditions, right, which result. These are rare, but certainly, they do exist. Likewise, there are germ cells which represent two different origin, right, and they are, germs, the next, generation, the child may not show 50% similarity. It will be much less than that, depending on which germ cell led to the formation of the kid, and so on. So this is a complexity which is very, very, unique, and we do see them, right. So that's what we pretty much looked at in Mendelian patterns. That is, whatever we discussed so far are for monogenic disorders, meaning you have phenotype resulting from defect in mostly one gene, right. But you also have conditions that are resulting from polygenic, meaning multiple genes interacting together and forming a particular phenotype, or the phenotype could be modified by the nature, which is called as a nurture. That's also, you call it as the epigenetics. Now, for example, obesity or diabetes could be genetic origin, but it also depends on your lifestyle. That the risk of you having a diabetes or obesity goes up depending on your food intake and many other lifestyles. So all the phenotype that you talk about today as of now could be a result of one of the three. That is, your genotype, your phenotype could be because of defect in one gene, or could be multiple genes, or could be environmental. What we understand now is that we can pretty much put a triangle something like this, connecting these three elements, single gene defects or multiple gene defects or environmental effect, and any phenotype that you look at can be placed somewhere in the triangle. If you talk about hand clasping, that your left thumb over the right, that is probably monogenic because you can trace it in the family. Hemophilia that we discussed, it is monogenic. But you have disorders, dominant disorders that are incomplete penetrant, meaning 60% penetrance. So you may have a genotype, but the probability that you would develop the disease is 60%. That is somewhere here. So there is some other gene that contribute which rescues from that, or complementation, for example. That again, it could be somewhere here, two genes rescuing each other. Diabetes, obesity can be genetic, environmental play. So you can pretty much place any phenotype that you are looking at in somewhere in the triangle. So that's the concept, and we will try to understand how we really dissect the genetics and genetic contribution to the phenotype, right. We will go on to discuss in the remaining lectures. So that's pretty much completes the second lecture for second week.